All right, so let's go ahead and move on to the second objective, which is describe what happens and the location of each step of transcription. All right. So now that we're moving on, it's important to ask the question, all right, so we have DNA. We know that DNA is like the instructions for everything in our body. Why do we even need RNA? Like, why not just use the DNA to make our proteins and make stuff in our body? And here's why. We know that DNA contains the instructions for protein, and that proteins are made by ribosomes. So we know the ribosomes are out here in the cell. That's where the proteins are made. The DNA is inside the nucleus. It's actually really risky or dangerous for the DNA to leave the nucleus. We gotta think about the DNA is precious. It's important that we keep it safe. If it gets damaged, then our body can no longer make proteins. We want to make sure that the DNA is safe inside the nucleus. But the ribosomes, they're really stuck inside the cytoplasm. It's not like the ribosomes can move to the DNA, where the DNA is. They're kind of stuck out here inside the cytoplasm. And so this is why we need RNA. RNA is the messenger. So think of like those owls in Harry Potter. They send messages back and forth. The owl, the RNA, the messenger RNA or mRNA, is a message that can be sent from the nucleus to the ribosomes. And this, so write down this information down here in your notes, that messenger RNA or mRNA. And once again, think of this image of the owls in Harry Potter. It's a message being sent from the DNA, from the nucleus to the ribosomes out in the cytoplasm. Okay. So there's another vocab word that you want to know. This is the vocab word of transcription. And transcription is basically the process in which nucleotides of DNA are copied into mRNA. And so here we have the DNA. We're using this DNA to make that complementary mRNA or messenger RNA. Make sure you're following along in the notes. So move on to the next page. Still on the right words for that. All right, great. So let's talk about how this process of transcription, of turning our DNA into RNA, actually happens. Okay? So the very first step is that we copy the DNA info onto the mRNA. Okay? And so what we're going to do is we're actually going to write in the corresponding RNA strand. So if this is our DNA, we know that T matches up with A matches up with U, A matches up with U. And we're going to end up with the RNA that complements the DNA. And so notice AT, AU, AU, GC, CG, TA. Very similar to matching up DNA when we do DNA replication. The big difference, remember though, is that there's those U's in the RNA. This is our RNA right here. This is our DNA right here. So very similar, but RNA has the U's. One question I get a lot from students is like, oh, but if there's a T in RNA, what does that match it up? A T still matches up with an A in mRNA. It's just the A matches up with a U in mRNA. So just know that difference when you're doing this as well. All right, you don't have to copy this all down. It's more just to get the general idea of which ones match up. The next step is now that we've made our messenger RNA, our mRNA, that little owl or that messenger is ready to leave, and so we have the DNA inside the nucleus. That messenger is now going to come out and go to the ribosome. So it's leaving the nucleus. I have this great animation I'll show you guys in a little bit that actually shows this whole process in action. But the mRNA is now going to leave the nucleus. Okay. Here's that animation I was telling you about. So here we have an enzyme. We're going to make the mRNA that matches it up. So notice how the G's and C's are matching up, the T's and A's, the U's and A's are matching up. And so now we have our mRNA that's ready to be sent out. Okay? So watch this animation. This is actually how fast it happens. So I'm going to start this actually probably at about 46 seconds. But this pretty incredible. This is actually the mRNA being made. And so you can see this red part is the DNA. This is the enzyme making the mRNA. And this big yellow strand, this is the mRNA being made. And it happens super fast. Like, you can see how quickly it's happening. And so this big long strip of mRNA is being made. And you can really notice that it's single-stranded here versus this dRNA is a lot bigger. It's that double strand. All right, so let's do a practice problem now. Which of the following statements is correct? So this is a key vocabulary word. We're talking about correct, not incorrect, correct. Concerning transcription. So that's another key vocabulary word in this question.
Where is it?
So if you need to, go back and check your notes. But read over which of these 4 statements is true or correct about transcription.
I think it's because we have different classes. We found that one's more advanced from the other one. They should be the same. Should be the same. They should be the same. What? The same class. People like the same procedure.
All right. So during the transcription, the adenine of DNA joins with the thymine of the RNA. We know this is not right because we know that actually if we're talking about uracil in RNA, there's no thymine or T in RNA, so we can eliminate that answer choice. During transcription, DNA is used to form a strand of mRNA. Okay, that seems like a possibility. Let's leave that as a possible answer choice. During transcription, both strands of DNA are transcribed at the same time. But that video we just saw, was it both strands being transcribed, or was it just one of them making that single strand? So we know that actually it's not both, it's just one strand. So this is not the answer choice. During transcription, one strand is the leading strand, one strand is the lagging strand. This is a what? I've never heard of this before. That's a clue. If you're getting vocabulary words that you've never heard of before, and you've really studied the playlist, that's actually a sign that that is not an answer choice, because if it was an important vocabulary word, we would actually give it to you. So don't fall into the trap of saying, oh my gosh, this is a vocabulary word I don't know. That must be the answer. This is actually not the answer. This one that we had before, B, is the best answer choice. And so be careful of falling into that trap of finding a vocabulary word that we've never seen before. That may be not be the right answer. All right, so we're done with the second objective. We're about to move on to the third objective. Just like last time, though, I want to turn this objective into a question that you answer. So it says, describe what happens in the location of each step of transcription. So let's make this into a question. What happens? Okay. And we want to know the location and in each step of transcription. Okay? So write down the location of each step of transcription and what's happening. If you need to, go back and check those notes. But I want you to turn this objective into a question and make sure you can answer this question.
Step 7. Biology. Step 7 of biology.
Where is it happening? What's this thing in the middle called? Yes, that's a good answer. Okay. So once again, we're turning this into a question. What happens and what's the location of each step of transcription? The location, if you remember, the whole reason why we need mRNA is that the location is the nucleus. This is the part of the cell where this is happening, and eventually that mRNA is going to leave the nucleus. But this is why we need mRNA in the first place, is that DNA can't leave the nucleus, so it has to happen in the nucleus. And during each step of transcription, the big thing that we're doing is we're making mRNA, and then the next step is that mRNA leaves the nucleus. those are the two big steps of transcription. We make the mRNA, we make that yellow snaky mRNA, and then that mRNA will then leave the nucleus.